Hello, this is Dr. Bell again, and this is the last lecture I'm going to give you, and it's on wound healing. There are two main sources for this lecture, and they are one is the chapter in the textbook on hemostasis and thrombosis, and the other is a book on oral wound healing that is available from the OSU library as an ebook. In this lecture, I'll first look at wound healing in general, including these stages of the process. And those will be hemostasis, inflammation, reepithelialization, granulation, and remodeling. I will then talk about aspects of wound healing that may come up in dentistry, including following tooth extractions for periodontal regeneration and techniques that involve tissue engineering. Here again are those stages of wound healing that occur in time. So I won't repeat them, but one point is that they occur in this general order, but there's some overlap between the stages. So reepithelialization may be occurring the same point as granulation and so forth, to some extent. And then the time scale for the entire process is usually weeks, which everyone has experienced, I think. Now, most dental procedures are going to involve healing of mucosal epithelia of the oral cavity, but most of the research on wound healing has been done on skin, and the mouth epithelium is actually a little bit similar to skin in terms of that both tissues are stratified squamous epithelium, as shown here. So the squamous epithelium has layers of cells that have a cobblestone appearance when looked at from the side, and they sit on top of a basement membrane, as shown here. Now, differences between the oral and the skin is that the oral epithelium, of course, is mucosal, so it's producing mucus that overlays it, and only some of the areas are keratinized, like the skin. So the skin is a keratinized epithelium where the outermost layer of cells die off and produce a layer of this protein keratin at the top in the diagram there, whereas only some of the oral epithelial do that. Now I'll start discussing the stages of wound healing and beginning with hemostasis. And so that is from the Latin meaning stoppage of bleeding. And there are two processes that combine to stop the bleeding. One is vasoconstriction and the other is blood clotting. Now, if you break down the clotting, there's also two processes that go into clotting, and those are the platelet aggregation and a fibrin clot that's formed from fibrinogen. So a final point about hemostasis is that it needs to be tightly regulated, because either too little or too much is bad for the organism. If there's too little, it can lead to hemophilia, but too much can lead to thrombosis or heart attack or stroke. Here's a view of the structure of a wall of a blood vessel, so we can look at some of these processes. In an injured vessel, the blood contacts this layer of endothelial cells. And they're making the molecules nitric oxide and prostacyclin, which help keep clotting down. And the layer that contains the epithelial cells is called the intima. There then is a layer called the media that has smooth muscle cells, which can contract in response to platelet thromboxane A2. And that can lead to the vasoconstriction effect of hemostasis. And I'll get into platelets just after this, so we can understand where that thromboxane A2 is coming from. there's and there are more smooth muscle cells in arteries than veins generally. So the vasoconstriction is stronger in the arteries. But finally, there's a layer of called the adventitia on the outside of the blood vessel that contains a few fibroblasts and also contains the proteins collagen and tissue factor. And normally the blood doesn't have contact with those proteins. But if the vessel is injured, there can be contact, and that's one of the sort of major stimulus that initiates the blood clotting process. So now I'll start in talking about platelets. The left-hand side of this slide shows the process of hematopoiesis, which makes the cells that are in the blood and is going on in the bone marrow. So it starts with the hematopoietic stem cell, or HSC for short, up top here, that can self-renew and also go to make up all progenitors that make up different lineages. They can make up white cells shown here, red cells here. And the lineage that we're interested in makes a cell called the megakaryocyte, and that goes on to make the platelets. On the right-hand side of the slide, then, what we can see is that the platelets are small pieces of the megakaryocyte cytoplasm and membrane that pinch off from those extensions. And they're not complete cells. They don't contain the nucleus, so they're just a little bag of membrane and cytoplasm, and they also have some cytoskeletal components and other organelles, as we'll see, but no nucleus, and so they're kind of a dead end. They can't reproduce themselves. They just go out into the blood and are continually produced and used up. When platelets are stimulated to participate in clotting, they undergo a process called activation. On the left-hand side here, you can see that one thing that happens is that they change shape radically. So normal circulating platelets are pretty flat and smooth, while the activated platelets, you can see, have a lot of protrusions due to extensions of the cytoskeleton into the membrane. And I think you can see that the altered shape is one element that makes them stick together more and be able to form the clot. And on the right-hand side is an activated platelet in a scanning electron microscope, and it's sitting in between a red blood cell on the left and a lymphocyte on the right. So you can see it's fairly small compared to those other cells. This shows signaling processes that occur in platelet activation and how they are affected by some drugs. So there are 4 receptors that can lead to activation here. And going from left to right, the first one is for ADP, the nucleotide. I showed you a bunch of nucleotides acting as signals within cells during the signal transduction lecture. Here's an example of extracellular signaling with a nucleotide, the adenosine diphosphate. It's activating a 7-transmembrane G protein coupled receptor, and you can see there are a number of drugs that interfere with this receptor, including clopidogrel, which is also known by the brand name Plavix. So that blocks the ADP receptor, and that's why it's an anticoagulant. Other receptors that can stimulate platelet activation are the adrenergic receptors binding epinephrine, receptors for collagen. Remember that as usually cells in the blood only see collagen if the vessel is damaged, and also receptors for thrombin. The thrombin receptor is interesting in the way it gets activated. Thrombin is a protease, as we will see in later slides, and it cleaves a 7-transmembrane receptor and exposes a new N-terminus of that receptor, and that N-terminus then stimulates signaling through the receptor. So it doesn't actually have a ligand; it just cleaves off part of the tail to expose the ligand that's already there. So one branch of signaling for platelet activation goes through eicosanoid lipid mediators that are made in the mitochondria from arachidonic acid. And if you remember, I mentioned them in the signal transduction lecture briefly as well. That can produce the thromboxane A2 that, as I mentioned earlier in this lecture, can cause smooth muscle contraction and vasoconstriction. That's part of hemostasis. It can also stimulate pathways within the platelet as shown here. You can see that the aspirin blocks the cyclooxygenase step of eicosanoid synthesis, which is why aspirin has its anti-clotting effect. Finally, there are a couple of membrane glycoproteins, or GP for short, that are activated to bind to extracellular matrix and aggregate the platelets together, and to make it form the platelet clot. And they're shown here. There's a glycoprotein GP1B complex with GP9A binding to von Willebrand factor, and through that to collagen, and then GP2B complex to GP2A binding to fibrinogen. And they're all leading to platelet aggregation. With activation, a few other things occur with the platelets. One thing is granule secretion. The platelets contain membrane vesicles called granules that have been divided into different types, and activation causes them to fuse with the plasma membrane and dump their contents into the extracellular fluid. And then there's also the morphology change I showed earlier, with the cytoskeleton changing to get, forming those protrusions. And there's a change in the membrane, and some of the lipids are flipped from inside to outside to make the surface more negatively charged than it was before activation. And the platelets aggregate to form a platelet plug. So in terms of these granules, there are different types. So there are little membrane vesicles within the platelet that it can, send to the extracellular when they get activated. And there are alpha granules that contain P-selectin, which is an adhesion molecule that can bind leukocytes, so it can kind of bring the white blood cells into the clot to make sure that, that they're fighting infection if needed. There's platelet factor 4, which is a signaling molecule, PF4. There's transforming growth factor beta 1, TGF beta 1, and platelet-derived growth factor, PDGF, which are cytokines and growth factors. And there are some molecules that are involved in clotting, including fibronectin, von Willebrand factor, fibrinogen, and the clotting factors 5 and 13. There are also a type of granules called delta granules that have that ADP that I mentioned that can, feed back and cause a positive feedback stimulation of platelet activation. And they also contain calcium and serotonin. And I'll talk more about calcium in a bit. So now we'll move on from platelets and get into the fibrin part of the clot, which is the second major part of the clot. In terms of the biochemistry, basically there are a set of clotting factors that I will show you that are identified by numbers and sometimes other names. They usually have an inactive and an activated form. Most of the clotting factors do. And generally, they're converted from the inactive to the active by proteolytic cleavage. The active form is often a protease itself that can cleave later proteins. So you get this cascade-like effect where one factor gets activated and then it activates the next factor and that activates the next factor. So the terminology for these factors is as follows. in the short, the abbreviation is generally F if it's for the factor, then Roman numeral and a lowercase a if it's the active form. So this FIXA is for factor 9A or activated factor 9. A few of the key factors have different common names, as shown here. Factor 1 is the same as fibrinogen, and its activated form is fibrin. Factor 2 is prothrombin, and its activated form, factor 2A, is thrombin. And tissue factor was formerly known as factor 3, although that isn't used anymore. There's an unusual bit of biochemistry that occurs in blood clotting, and that is that they contain a modified amino acid, gamma-carboxyglutamate. So it's an amino acid that is not one of the 20 usual ones that can be introduced by the genetic code. And the factors that are modified in this way include thrombin, factor 7, factor 9, and factor 10. And gamma-carboxyglutamate is made post-translationally from glutamate. So it's on a glutamate that's already incorporated in the protein is changed to gamma-carboxyglutamate. The process for making it involves this vitamin K. So if the organism is deficient in vitamin K, its blood clotting doesn't work properly. The modified amino acid gamma-carboxyglutamate can bind calcium ions, and calcium is required for clotting. And one way that we know this is that for instance, the purple cap blood collection tubes that you may have run into, they contain a compound called EDTA that prevents clotting by chelating the calcium ions. So if no calcium, the blood doesn't clot. And that's why those purple tubes work. And the process of modification is blocked by some anticoagulation drugs called coumarins. And warfarin is the best known of those kinds of drugs. So if you block this gamma-carboxyglutamate, you can actually block blood clotting. And that's used pharmaceutically. So here are the details of this how this reaction works and how vitamin K gets involved. Up here, we're looking at the glutamate residue. You can see it's indicated that's part of the peptide chain of the clotting factor because of the dots on either side and then you see the side chain hanging down from the peptide chain. And you can see there's a carboxylase enzyme that uses CO2 and oxygen and reduced vitamin K and produces vitamin K epoxide, protonated water, and the gamma-carboxyglutamate side chain in the protein. And you can see why it binds calcium, because it's got two negative charges there that interact well with the two positive charges in the calcium ion. And the second view shows what is going on with vitamin K, and this is important to what the coumarin drugs are doing. And this part of that diagram is the same reaction that is on the left-hand side diagram, but from a different point of view, where we're looking at the vitamin K instead of the gamma-carboxyglutamate. And you can see that the vitamin K is forming into the vitamin K epoxide. And so from that side, it's an epoxidase, along with the carboxylase activity. And then you see that the vitamin K gets recycled back into the form that can be used by the enzyme to make gamma-carboxyglutamate by two reduction steps. and it's those reductases that are inhibited by the coumarins, and so that's how coumarin is interfering with the blood clotting. So this then shows the coagulation cascade that leads to the fibrin clot. And it's obviously very complicated, and I'm not expecting people to know the full details of every factor in there or anything, but I'll try to hit a few of the key points. First, just in general, there's a progression of factors becoming activated and activating the next step, and you can see that for multiple times in there. And there are two ways that the pathway that fibrin clotting can be activated, two different ways, and one's called the contact or intrinsic pathway, and the other one is called the tissue factor or extrinsic pathway. And those two pathways, though, come together in that they both make activated factor 10, factor XA, and together with factor 5A works with that, and 10A is really the protease, and 10A then converts the prothrombin to thrombin, and that's really a central step in the fibrin clot. And it's a central step because the thrombin is then what converts fibrinogen to fibrin, which actually is what makes the clot. And you can also see those green arrows coming out of thrombin, where it actually can do a lot of different proteolytic reactions. In some cases, it's converting some of the earlier factors in the cascade, so it's kind of doing a feed-forward activity, but it also stimulates some inhibitory activities where down by thrombomodulin there, so it also has feedback. So it's very complicated what it's doing. And remember also, thrombin acts to activate that unusual G-protein coupled receptor on platelets to activate them. So it's cross-talking to the plate with pathway as well. So thrombin is a very central molecule. And then just a couple more things to point out here. The most common forms of hemophilia affect factors 8 or 9. And both of these genes are on the X chromosome, so they are much more likely to affect males because males only have the one copy of the X chromosome, so a single mutation can knock out these factors and cause hemophilia. Lastly, there's this protein called antithrombin that is a protease inhibitor of thrombin, and it's activated by the polysaccharide heparin. And that is why heparin has anticoagulant activity. Now to look at what fibrinogen and fibrin are doing, this is fibrinogen. It has 6 subunits. It has 2 each, alpha, beta, and gamma, and it has this long rod-like structure that all the 3 subunits are forming a triple helix to make the rod part of the rod. And it's made in the liver and is circulating in the blood. It's soluble as fibrinogen. Thrombin then first clips off a small peptide that is shown in red called fibrinopeptide A, and that's from the N-terminus of the alpha subunits, making fibrin 1. And that allows the ends of the rods to associate with the middles of other rods, as shown here. Once that FPA is removed, that association can start to happen, and so the rods kind of line up and form these linear structures that are called protofibrils. And thrombin also clips fibrinopeptide B then from the N-terminus of the beta subunits, making fibrin 2. And what that does is that releases a part of the alpha subunit that had previously been bound to the middle of the rod. And then that part of the alpha subunit can stretch across from one protofibril to another, and they start to bind together and cross-link between the protofilaments, and then it together it forms a large structural gel that can be the clot. So there is a way to reverse even after you've made a fibrin clot, and it's called fibrinolysis. So fibrinolysis is the cleavage of fibrin that can lead to dissolution of the clot. And it's carried out by an enzyme plasmid. And again, plasmid is synthesized as in an activated form, inactive form plasminogen, which is cleaved by plasminogen activators. And two of the important ones of those are TPA, which is short for tissue plasminogen activator, and urokinase, which is actually not a kinase, but it's a protease. But it's was originally purified from urine. And recombinant tissue plasminogen activator or urokinase are used as treatments for clotting diseases such as stroke. So they're known as the clot buster drugs. So we're going to move on from hemostasis and start talking about the what happens after the clot is formed and actually getting into how the wound gets healed. And I'm first going to talk about inflammation. And so inflammation has these characteristics, which you've all heard of the macroscopic characteristics of pain, heat, redness, and swelling. And so it's a symptom that can be found in many different places. And microscopically, inflammation is usually accompanied by an infiltration of leukocytes, white blood cells of various types, into the tissue from the bloodstream. And it can be a normal response, or as in wound healing, or if it gets out of control, it can cause a disease process. Now, after wound healing, inflammation can get started in a few different ways. One way is from the cytokines can get made by cells in the tissues that are injured, or as we've seen, the platelets contain some pro-inflammatory cytokines in their granules that come out during the clot formation. And some of those sometimes signals can come from cellular damage that will cause this, or it could be the presence of foreign microorganisms can lead to initiation of inflammation. And one cell type that can be involved in this is there are some immune cells that are always present in tissues, including our skin and oral epithelia. And so there can be macrophages that are always there. So sometimes macrophages monocytes in the blood can migrate and turn into macrophages in tissues, but there also are macrophages that are always around, even in healthy tissue. There are dendritic cells, which are cells that are specialized to present foreign antigens to the lymphocytes and cause an adaptive immune response. There are mast cells in tissues that are involved in allergic type responses, which are in terms of what they're supposed to do in healthy is to resist certain kinds of parasitic infections. And there are these cells that are present in tissues, the gamma delta T cells that are, kind of a new cell type that's being more studied, but they aren't the normal T cells that are involved in the adaptive immune response, but appear to be more of a bridge between the innate immune response to any foreign invader versus the adaptive immune, which recognizes a specific and has the invader and kind of has a memory for it. And if you look at inflammation, generally there are waves of white blood cells moving into tissues, and the simplest way is that there are the neutrophils are the earliest cells to arrive. Monocytes and differentiating into macrophages are the second wave, and lymphocytes are the last cells to arrive over time in inflammation. And to go into the functions of those cells, then the neutrophils, also known as PMNs for polymorphonuclear leukocytes, and basophils and eosinophils, which are related, are kind of the first responders of, as we've seen, they're the first ones to migrate. They do a lot of phagocytosis of foreign organisms, or they have granules that they can use that have antimicrobials, or they can generate reactive oxygen species. So they've got a number of ways of just fighting, especially bacteria. And then the macrophages are more involved in organization and regulation of inflammation and of the immune response. So macrophages are formed from monocytes that are in the blood. They also do some phagocytosis, but they produce a lot of cytokines and sort of help to modulate the response and organize it. And then the last wave, the lymphocytes, are in this adaptive response where they are going to generate a specific response to specific foreign organisms that are present, and can generate a memory so that they can respond faster if they see that organism again. And there are the two branches, the T cells that are involved in regulation of responses, and also can a cytotoxic response where they kill off the host cells to remove infections, and then the B cells, which are involved in antibody responses. This slide shows the process by which the white blood cells move into tissues during inflammation, and it's called extravasation. So first, the resident immune cells in the tissue, such as macrophages or mast cells, can sense either cell damage or the presence of foreign organisms by sort of standard, patterns on foreign organisms. And then they can release signals such as histamine, TNF alpha, or leukotriene B4, or these chemokines like interleukin 8 and those other ones that are listed there. These signals can affect the endothelial cells of blood vessels and cause them to fuse vesicles called Weibel Palade bodies with their cell surface. And that moves some adhesion molecules to the surface of the endothelial cells that leukocytes in the blood bind to. Initially, they bind to some proteins called selectins that we also saw were in platelet granules as well. And that binding is pretty loose, so that the leukocytes roll along the surface with the flow of the blood, even as they're bound to the selectins. But eventually, they engage stronger adhesion proteins called integrins, and that causes them to stop and flatten out against the side of the blood vessel. And eventually, they carry out a process called diapedesis, where they squeeze between the endothelial cells and move out of the bloodstream into the tissue. And from that point, they migrate toward higher concentrations of these chemokines, and that's called leukocyte chemotaxis. And to get to the area of the tissue where there is, infection, they just follow the gradient of those chemokines that are diffusing out from whichever cells are making them. the CXRs and CCRs that are listed there are chemokine receptors for different families, the CXC family and the CC family of chemokines. So just to mention about cytokines, they're secreted proteins acting in cell communication and frequently for immune responses. Interleukins are abbreviated IL with number, and there are a bunch of those. I think over 20 now known, and they're made by leukocytes and affect them and other cells. Interleukin sort of implies they're communicating between leukocytes. Then their growth factors can also be cytokines and usually have a GF at the end. They have at least the effect of stimulating cell division, although they often have other effects as well. Then these chemokines are structure related, small, and they cause chemotaxis of the leukocytes, and so you might see there could be things like CCR as the receptor and CXCL would be the ligand of a CXC family, but they also sometimes have names like interleukin 8 that is just a historical type of thing. And there's a number of cytokines that are pro-inflammatory and seem to be often be made in situations of inflammation. And examples of those are tumor necrosis factor, we've mentioned a couple times. Interleukin 1 is another one. Interleukin 6, interleukin 18, interferon gamma, and there are others. And then there's some anti-inflammatory cytokines as well, and that the most prominent one is transforming growth factor beta. So there is inflammation occurring in wound healing. It probably can reduce the risk of infection because you're stimulating immune cells to come in there and neutrophils to clean out the bacteria, and then lymphocytes to generate antibodies and so forth. So that's however, in gene knockout studies in mice showed that inflammation is probably not absolutely required for wound healing. The actual wound healing part can be carried out without inflammation, and in fact, increased inflammation during wound healing can be associated with either the formation of larger scars or possibly even chronic wounds that don't heal. And oral wounds actually have less inflammation and scarring, which is similar to when people have made wounds in the fetuses of animals like mice. They have less inflammation and less scarring during their healing. So now I'm going to move on to what happens post-inflammation in wound healing, and a couple of steps that are going to happen. One is reepithelialization. That involves a cell type called keratinocyte stem cells. So they proliferate, they migrate, and they layer over the the blood clot, and create a new epithelial layer. And then there's processes with fibroblasts underneath the epithelial layer where they proliferate, they migrate into the clot, and they form something called a granulation tissue. Here's an example from the oral cavity of reepithelialization and the formation of granulation tissue. And this is a wounded human gingiva after 3 days on the left and 7 days on the right. And below is a diagram showing which cells are where in the picture above. You can see on the left 3 days, you can see the fibrin clot labeled with FC, and it's exposed to the surface of the mouth still at that point. And the epithelial layer is shown by E, and there's some proliferating and migrating keratinocytes at along the base of the epithelial layer further in, and migrating into the clot, or above the clot on the left. Then at 7 days, the epithelium has reestablished over the area, and the clot has turned into granulation tissue, which is marked with GT, and you can see the kind of granular appearance there of the granulation tissue. So that's caused by the granulation tissue is created by fibroblasts migrating into the clot and putting in extracellular matrix, and Dr. Mukshan gave a lecture on extracellular matrix, and so it's these molecules that are deposited outside of cells in form like basement membranes, connective tissue like tendons and so forth, but it's also underlying the epithelial layers, and there are novel forms of extra ECM molecules are found in wounds. The fibrin in the clot is considered to be a form of extracellular matrix. It's outside the cell. It's deposited, and cells have to migrate through it and around it. And then there's something called fibronectin that comes in and attaches to the fibrin from the plasma, and is used in wounds. And then the cells deposit these other forms, laminin 332, tenation, and fibronectin EDA in the wounds. And then the connective tissue in the wounds is also cleaved by proteases in some cases to aid during healing. So the plasmin that I talked about in terms of fibrinolysis cleaves the fibrin and lets the keratinocytes migrate into the fibrin clot. And then matrix metalloproteases are also important in this. And various growth factors can cause proliferation and migration of the keratinocytes that are reepithelializing. Among those are epidermal growth factor, transforming growth factor alpha, keratinocyte growth factors 1 and 2, and eventually the epithelium gets completed. It completely covers the wound. New basement membrane is formed. And if this process fails, it can lead to chronic wounds that are always open. And examples of chronic wounds are diabetic ulcers, certain wounds that get burns, and other things that get bacterial infections sometimes end up as chronic wounds. And the dry socket following tooth extraction is the dental example of a chronic wound. So in the granulation tissue, then what happens? It restores the blood supply. Blood vessels grow back and have been disrupted, but they can grow back into the area. And the extracellular matrix deposited as part of the connective tissue and increases the mechanical strength and function. But it can also result in scar formation. If you only injure the epithelium and let that re-heal, you don't get a scar. So the scar is formed by this connective tissue underneath the epithelium, and that can have aesthetic defects or possible loss of function, fibrosis from too much scar formation. So the fibroblasts are the major cell type that are making granulation tissue and depositing extracellular matrix. They proliferate in surrounding tissues and migrate into the wound. They synthesize the extracellular matrix, and they can do this function where they differentiate to something called a myofibroblast, which is when they become almost like a smooth muscle cell. They develop a lot of the actomyosin cytoskeleton, and then they can contract to actually make the wounded area smaller and draw the healthy tissues closer together. And so how do these fibroblasts get activated? So here's a diagram. You can see the epithelium. You can see where the wound is with the blood clot there, and the connective tissue underneath the epithelium. And those green cells are the fibroblasts inside the connective tissue. What activates them to divide and migrate into the wound? It could be factors that are released from the extracellular matrix in the connective tissue. It could be inflammatory cells, either ones that came in through inflammation or were activated, were tissue resident but got activated. It could be keratinocytes, neuronal cells, adipose cells can release, various compounds in response to the injury, cytokines, chemokines, growth factors, and so forth that can activate the fibroblasts. Altered mechanical tension can actually activate them, or disruption of cell-cell contacts, reduced oxygen tension due to the blood vessels being disrupted, can activate plasma-derived factors, or proteins present in the blood clot. We've seen how platelets, use out of their platelet granules, they release some substances, and then it could be microbes coming in from the saliva that activate the fibroblasts. The growth factors that are causing proliferation, migration, and extracellular matrix synthesis can include fibroblast growth factor 2, epidermal growth factor, platelet-derived growth factor, and we saw how that came from the platelet granules during clotting, and insulin-like growth factor. To talk about this contraction of wounds, then, there are the myofibroblasts that I mentioned form from fibroblasts. They resemble smooth muscle cells. They have increased actin cytoskeleton, and in fact, they have the smooth muscle actin isoform instead of the cytoplasmic actin isoform. And they contract like muscle and reduce the size of the wound. There's connective tissue remodeling that occurs during the process. The initial connective tissue that's deposited is disorganized, and it gets more organized, and the extracellular matrix molecules are replaced by more mature forms. For instance, type 3 is replaced by type 1 collagen. There's alignment and cross-linking of collagen fibers that occurs. And by this process, the breaking strength of the wound increases. So over weeks and months, you can return back to up to 80% of the normal tissue breaking strength. So angiogenesis is the process of growing new blood vessels into the wound. They're needed because of disruption of the existing ones. Their endothelial cells are normally quiescent, but with stimulation, it can cause sprouting of blood vessels. And that involves vascular endothelial growth factor, or VEGF, or VEGF, that responds to hypoxia. And I mentioned in the gene expression lecture that involves this hypoxia-inducible factor 1 alpha that is regulated by oxygen and is turning on the gene for vascular endothelial growth factor. And just as a side note, solid tumors need blood supply, and they often cause the growth of blood vessels through this vascular endothelial growth factor. And there are some tumor therapies that are trying to target this and deprive the tumors of their blood supply by inhibiting the VEGF signaling. So now I'll talk some about bone healing. Normal bones are continually remodeling. There are osteoblasts and osteoclasts. The bones are made of hydroxyapatite and also collagen. Hydroxyapatite is that calcium phosphate compound. And there are cells present in the bone. After an injury, there's a recruitment of cells called mesenchymal stem cells, MSCs. They get recruited by a chemokine that's called stromal cell-derived factor 1, or CXCL12, and it binds to CXC chemokine receptor 4 on the mesenchymal stem cells and attracts them into the area of injured bone. So SDF1 binding to CXCR4, and they can differentiate into osteoblasts. Bone deposition occurs with collagen first, followed by the hydroxyapatite, and involves bone morphogenetic proteins, BMPs, especially BMP2. And the bone needs to be vascularized as well, which involves this vascular endothelial growth factor. And then there's remodeling of the bone as it is deposited. The stages are distinguishable by their microscopic appearance. The first stage is woven bone that's laid down along disordered collagen, and a later stage and a stronger stage is the lamellar bone that is layered along oriented collagen. Directed wound healing is a key thing in dentistry. And that's because regaining specific structures is important. You want to have bone grow back where there's bone, and in the periodontium, you want to have the tissues grow back in their right place. So there are techniques to try to manage this that are under the name tissue engineering. Some techniques involve membranes that can block unwanted cells from entering an area that's healing. Those membranes are often of a form that's resorbable, so they will block for a specific period of time, but the body will eventually remove them. There can also be substrates that are used that act as scaffolds for cells and try to create the type of healing that is wanted. Or growth factors and biologic agents can be used. I've listed off a lot of growth factors used in various, the bone morphogenetic proteins and so forth. And there are specific surgical techniques and flat placements that might be used to try to guide the healing in the correct direction. This shows the time course of wound healing following tooth extraction. The tooth is extracted first. The clot forms fairly rapidly within a day. And then within 2 weeks, granulation tissue forms in the socket. By 4 weeks, there's provisional matrix has been made by the granulation tissue. Then after 12 weeks, you get a transition of woven bone into the lamellar bone, the stronger later version. And after 24 weeks, it completely heals up and fills in the socket. One possible problem with extraction healing is that the ridge of bone may not heal to the full width that was there when the tooth was present. And here you can see an example of that with the reduction of the alveolar ridge width after 12 months following extraction. So as what happens during socket wound healing? The bone progression works as it does for fetal bone development and for fractures, which is this progression from woven bone to lamellar bone. The extent of bone can be less than the original, as is shown in that picture. And the ridge width especially can be less, that's especially for the buccal side and with molars that the ridge width becomes a problem. It can make implant placement difficult then if you wait for the extraction to heal before placing the implant. It can be hard to have enough bone to place the implant. So possible interventions for this are immediate implant placement as a possibility, or using graft materials such as hydroxyapatite or polylactic co-glycolic acid to try to fill in the extraction socket. Barrier membranes can also be used, or biologics like the bone morphogenetic proteins that we've heard about can be used to try to stimulate the bone growth and keep the ridge width high. This is an example of a treatment used to try to use immediate implant placement following extraction of 2 teeth and to try to get full width healing. And so in the first slide, you can see that the teeth have been extracted, and then the implants are placed in the sockets. And in one case, there's like no room. There's no bone left out to the side, so you can see down the side of the implant. They used in this case a deproteinased bovine bone mineral called BioOs to fill in the area, and then covered it with a membrane made of resorbable collagen. So on the left here, you can see the scaffold material being placed in the socket, and then the resorbable membrane covering it. And then they allowed that to heal and went back and can see in the bottom panel that the bone has filled in nicely around the implants in that case. And there's still, it's still somewhat up in the air whether immediate implant placement is better in all cases or not, or there's also early implant placement, or the older way was definitely to delay the implants until full healing, but all three are undergoing. And I've seen some, if anyone's interested, I've seen articles on comparing the 3 methods. Now, another way that this tissue engineering on wound healing might be used is following periodontal pocket reduction surgery. Here's an example of a tooth where on the right side it has the yellow biofilm of bacteria that's caused the pocket to be deeper, and the bone is recessed and normal chronic periodontitis, and the periodontal ligament is lower, and the left side of that tooth is healthy. And in periodontal surgery, you might pull back the gum and scrape off the tooth and clean off the bone and so forth, and religate it and hope it heals properly. But it's also possible to use techniques to try to guide the tissues. So here we can see on the left-hand side a diagram of a periodontal pocket where you can see that there are different tissues that can be sources of repopulating stem cells. They could come from the epithelium, number 1. They could come from the connective tissue, number 2, from the bone, number 3, and from the periodontal ligament, number 4. And so on the right-hand side, you can see there's a membrane been placed to try to make it so that the lower parts are repopulated with stem cells from the bone and the periodontal ligament rather than from the epithelium or connective tissue, and try to get restoration of the bone following the periodontal surgery with a membrane approach. Here's a large chart that shows a lot of the different tissue engineering approaches that can be used in dentistry. And they've divided them up into three major categories. You have the scaffolds, so the solid materials that are trying to act as a place for the tissues to grow right, and they can be all these different materials, composites of materials, and then delivered in different ways, possibly. Another component can be cells, and stem cells are a very hot area of research. And I mentioned mesenchymal stem cells earlier in the lecture. They're one of the stem cells that people are looking at to try to use in dentistry, or various other kinds, and try to generate. The mesenchymal stem cell can go to be an osteoblast, and that's shown there. And then there can be various transcription factors or matrix proteins to try to use with the cells. And then finally, there's genes and proteins, some of which we've heard about, that you might try to use to growth factors and differentiation factors that can help the process of wound healing. They sometimes, or oftentimes with those, a single administration is probably not going to be effective. These, proteins are not that stable, so they might be put into gels or microparticles to try to release them slower, or it might be possible even to use gene therapy to introduce growth factors and differentiation factors that stimulate wound healing. And then combining all three of those approaches, it might be possible to really get the final approach to generate desired wound healing. And here's some compounds and combinations of compounds that are in clinical trials, and this is not too recent now, but we can see here that molecule PDGF, platelet-derived growth factor, is one thing that's being looked at. It can cause proliferation of fibroblasts and osteogenic cells, along with a scaffold as well. Or here's the fibroblast growth factor 2 being used with another scaffold in this clinical product. And then there are various others listed there too. So, and the healing around implants actually seems to be pretty well solved in terms of the bone. They've achieved good success in changing the implant materials so that the bone heals well into the implant, that's known as osseointegration. And so the soft healing around implants now has become more of a focus of attention in aesthetics outcomes. And so that's where I'll leave it with. And I'll just go back and recap some of the things I've talked about in this lecture now. So under hemostasis and thrombosis, there are two components to blood clots: the platelet clot and the fibrin clot. Platelets are the cellular fragments from megakaryocytes. They're activated when the blood vessels are damaged. They have membrane around them. They have these granules, which are the membrane vesicles inside them that contain various things. Fibrin is produced by a cascade of reactions involving these clotting factors, and thrombin is a central factor in that. And fibrinogen changing to fibrin actually makes the clot form. And then there are various pharmaceuticals or genetic diseases that affect blood clotting. And aspirin affects the eicosanoid pathway in platelets. Warfarin affects the vitamin K pathway that's involved in gamma carboxyglutamate modification of clotting factors. Plavix is affecting the ADP receptor in platelets, and heparin is affecting antithrombin. And hemophilia and von Willebrand disease affect various parts of blood clotting. I didn't really mention von Willebrand disease, but it affects that von Willebrand factor that I mentioned briefly. And hemophilia affects the two clotting factors. And finally, there's fibrinolysis to remove the clots. That involves tissue plasminogen activator or possibly urokinase activating plasmin to plasminogen, or plasminogen to plasmin, and the plasmin proteolyzes the fibrin and can remove the clot. We talked about inflammation, that white blood cells enter tissues by extravasation. There's a defined order of neutrophils, monocytes, then lymphocytes in terms of which cells are entering tissues over time. Cytokines and chemokines are involved. There's some pro and anti-inflammatory, generally, cytokines. And in terms of reepithelialization and granulation tissue, the keratinocytes make the epithelium. Fibroblasts make the granulation tissue, and they go on to produce connective tissue. And growth factors and cytokines, again, are involved. The myofibroblasts are derived from the fibroblasts and can contract the wound to make it smaller. Now about remodeling and specialized cells in bone. There are osteoblasts and osteoclasts. It's constantly remodeling. Bone morphogenetic proteins stimulate the differentiation into osteoblasts. It forms woven bone and lamellar bone, both during development and during healing of fractures and defects in bone. There's angiogenesis is the process of restoring blood vessels to wounded areas. The endothelial cells grow out, are stimulated to divide, although they're normally quiescent, and that involves vascular endothelial growth factor. And applications to dentistry, many general wound healing concepts can apply to dental situations. Many of the same cytokines and stuff are involved, and the situations can be extractions, periodontal regeneration, dental implants. There are special circumstances in dental wound healing. Bones are often involved, or the periodontal ligament is a special situation, and the tooth or implant can be close up to the tissue that's trying to heal, and that can sort of affect which cells want to migrate into which area and so forth. So finally, there are these techniques that are used to guide wound healing in dentistry. Among them are membranes, especially resorbable membranes of various types. These substrates that can be used to help the cells to populate and grow, and established, and growth factors can be used, and stem cells can be used potentially. And that's all I have for today. Thank you.